In my previous lecture, I talked about the endocrine system, that is how the hormones are being secreted and what is the function of different kind of hormones. So after talking about the endocrine system, now I will be talking about the animal nervous system in this particular lecture. So the nervous system consists of neurons and supporting cells. So now these neurons or supporting cells, they could be categorized into three different categories. So number one is the sensory or the efferent neurons. So these neurons, they carry impulses from sensory receptor to the central nervous system. Now, these neurons, they have fine branches called as dendrites, which are profusely branched so as to collect a lot of stimulus. Second one is the motor or the efferent neurons. They usually carry impulses from the central nervous system to the effector muscles and glands. So, in response to the stimulus, the body needs to do some kind of activity. Let's say, for example, the hand, if it is moving towards the heat, or let's say moving towards the fire. So, after sensing the high temperature, it needs to be immediately removed back. So that function will involve sensation or the sensory neurons action and followed by the motor neurons action. So the hand will be prevented from being burned. Now the third one is the associated neurons. They are found in the brain and spinal cord and often provide link between sensory and the motor neurons. So here you can see example of a sensory neuron. So here we have find dendrites which could be present at the site of the tissues or body parts. So let's say if we talk about the skin, so there should be a lot of dendrites just placed in the areas where the sensation is required. Let's say below the skin. So that could lead to the follow of direct impulse to the cell body. Now it could be connected by the associated neurons which are in the part of the brain or spinal cord, and ultimately that associated neurons could be connected to a motor or efferent neuron, which is again taking the impulse back from the associated neurons to the motor neuron where the action needs to be done. Okay, so the vertebrate nervous system can majorly divided into two divisions. That is the central nervous system and the peripheral nervous system. So now the central nervous system consists of two major components, that is brain and the spinal cord. The peripheral nervous system consists of two major components, that is the sensory pathways and the motor pathways. Now these motor pathways can further be divided into voluntary nervous system and the autonomic nervous system. Also, this autonomic nervous system can further be divided into sympathetic and the parasympathetic divisions. So this is a brief outline of what the nervous system different components are and what it consists of. Okay, so let's see the structure of a neuron. So the neuron majorly consists of a swollen or enlarged structure which is called as the cell body. Now this cell body consists of a nucleus, and from this particular cell body are a lot of protruding structures or small fiber-like extension called the dendrites. Now these dendrites could further divide into fine dendritic spines. These dendritic spines, they are fine fibers or thread-like structures which are usually meant to increase the surface area of the dendrites so as to receive stimulus from a large area. So usually a single axon could be protruding from the cell body, which is responsible for the transmission of the impulses from the cell body to the later part. Now this axon could have a myelin sheath which could facilitate in rapid transmission of impulse. Now this myelin sheath, if we look into a little bit detail, so we can have intervals or interruptions between the myelin sheath, which is these gaps which are known as the node of Ranvier. So in the peripheral nervous system, the myelin sheath is formed by the Schwann cells. So if we take the transverse section of the area where the myelin sheath is present, so we can, it appears something like the axon is inside and outside we have a dark myelin sheath. So the neurons are supported both structurally and functionally by supporting cells which are called as the neuroglia. Now these neuroglia cells, they are almost 10 times more as compared to the neurons. So these neuroglia cells, they are in abundance. They are present in large numbers. They have a lot of functions. So some of the important functions are that they supply nutrients to the neurons. They are also responsible for removing the waste products from the neurons, guiding the axon migration, and providing the immune functions. There are two types of neuroglia in vertebrates. These are the Schwann cells and the oligodendrocytes. So they, both of these Schwann and the oligodendrocytes, they produce myelin sheath that usually surrounds the neurons. Now the Schwann cells, they produce myelin sheath in the peripheral nervous system, while oligonucleotides, the oligodendrocytes, they produce myelin in the central nervous system. And in simple terms, those axons which has myelin sheath covering onto their surface, they are known as myelinated. And those axons which do not have this myelin sheath, they are called as unmyelinated. So this is how a myelin sheath is formed. So usually onto axon, the Schwann cells, they wrap themselves around the axon in multiple layers. They are usually in several layers to form the myelin sheath. So in the central nervous system, myelinated axons form the white matter, and the unmyelinated dendrites and the cell bodies form the gray matter. In the peripheral nervous system, both myelinated and the unmyelinated axons, they are bundled together to form nerves. Okay, so let's talk about the how the impulse are moved from one point to another. So there is a term which is called as the resting membrane potential. So now, as all of us know, that the neurons, they usually communicate with one another via change in the electrical properties of the plasma membrane. That is, usually there is change in the electrical potential or the surface electrical properties that are responsible for the movement of electrical signals, which are known as impulses. Now usually the inside of the cell is more negatively charged as compared to the outside. Now there are three important reasons for that. So number one is that inside the cells there are large proteins, nucleic acids, and all of these carry high negatively charge. Now, because of the abundance of these proteins and nucleic acids, the cell has high negative charge. And this high negative charge is known as the fixed anions. So these molecules, frictional ions, they impart large negative charge. Now, second one is the sodium potassium pump that brings two potassium ions inside the cell and pumps three potassiums out. So it appears like if you balance, three potassium is two potassium coming out and three sodium moving out. It means there is loss of one negative charge, so one positive charge. So that imparts one extra negative charge to the inside of the cell. The third one is these ion channels. They allow diffusion of potassium. So potassium can easily diffuse through the ion channels. So that will lead to decrease in the positive charge inside. So it means that there is more and more increase in the negative charge as the positive charge cations or the potassium is moving out. So the sodium and potassium channels in the plasma membrane, they pass through the gates. There are a couple of gates to open or close the channel pores. So in the axons and in muscles, these gates they open or close in response to the membrane potential. So these are known as voltage gated ion channels, since they are operating on the basis of the impulse being received. Okay, so let's have a look at the sodium potassium pump. So this is a pictorial representation of a sodium potassium pumps. So this pump usually transports three sodium molecules outside of the cell. Sorry. So three sodium they can be transported outside, and in this process there is a one ATP being utilized, it is converted to ADP. And also the two molecules of potassium, they can be pumped inside. And this whole process requires utilization of energy. So when there is no stimulus, so there is no impulse being received by the neurons. So at that particular time, the neurons have a charge which is known as the resting membrane potential. Now, this resting membrane potential is about 70 millivolts, which are also known as 0.07 volts. So this is a typical representation of a voltage gated ion channels. So in neurons and muscles, there are channels especially meant for sodium and potassium. So at the resting membrane potential, so these gates are usually closed. Now, when there is a threshold level of depolarization attained, so threshold means that at a certain level after which the activity starts. So let's say, for instance, let's say there is a bike or a car which is just lying idle. So now if somebody starts to push, then there's an amount of force that is required to move the car or the bike. So that is the threshold amount of force. It is not that somebody just touches and doesn't even apply strong force, it starts moving. So that level of force which could move the vehicle is known as the threshold force. So similarly here also, until unless a threshold level of charge is attained or a potential is attained, these voltage gated channels will not open. So the positively charged ions, called as the cations, they are outside the cells. They are attracted to the negatively charged ions which are inside the cells. So the resting membrane potential is more permeable to potassium than to other. So it means the potassium enters the cell. So in that now we have the activity of sodium potassium pump. So this sodium potassium pump is driven by a electrical gradient. So when there is excess of potassium inside, it will be driving the potassium out. So now there are two forces which are acting at one point of time. So what is that? That at this potassium by a electrostatic interaction, or you can say attracted towards the negative charge inside cell, will try to move in. However, the sodium potassium pump will try to move them out. So at some point, there will be a balance between the influx of potassium and efflux of potassium. So this is known as the equilibrium potential. So for potassium, the equilibrium potential is minus 90 millivolt. So if, let's say, the equilibrium potential is minus 80, so at that particular time, it means that there is a difference of 10 units. So to now bring it to minus 90, potassium will move out. Now in other case, if there is increase in the minus value, that is from minus 90 it becomes minus 100 millivolt. So at that particular time, the potassium ions will diffuse inside the cell, so as to again maintain the equilibrium potential to minus 90 millivolt. So this is a representation of different kind of ions. So this is the concentration in the cytoplasm. This is the concentration in the extracellular fluid, the ratio and the equilibrium potential. So sodium in the cytoplasm it is 15, outside is 150. So if we take the ratio, it is usually 10:1, and the equilibrium potential is plus 60. For the potassium, 150 millimolar inside the cell, 5 millimolar outside. The ratio is 1:30, and the equilibrium potential is minus 90. For chlorine, inside it's 7 millimolar, outside it is 110. The ratio is 15:1, and the equilibrium potential is minus 70. So this equilibrium potential they are usually maintained, and if they are disturbed, then the condition is that there is movement of ions, or depolarization and repolarization is taking place. So if the depolarization is there, then the sodium potassium pumps, or they will be coming into action. The energy will be utilized again to maintain the equilibrium potential. Okay, so let's have a look at the action potentials. So there could be small change in the membrane potential. They are called as the graded potential, because their stimulus is not that high that it could lead to the movement of a impulse. It is just small fraction of potentials. So let's imagine that this is a membrane potential that we are measuring. So this is the membrane potential minus 70 millivolt, which is at rest. Now, if there is a very weak excitatory signal E1, so that could elicit a very small response. Now, after a gap of few milliseconds, there's another one which is a little bit better as compared to the first excitatory signal. Now it could be very close that now we can have a inhibitory signal. But however, this is kind of opposite to what we have the excitatory. Now these three graded potential, since they are very close, the time gap between these three potentials, that is two excitatory and one inhibitory, is very less. So in that particular condition, or at that particular time point, these can be added. So the overall potential, or the overall stimulus, will be the algebraic sum of these three signals, or three potentials. So E1 plus E2 plus I. So once a particular level of depolarization is reached, a nerve impulse or action potential is produced. So usually it is about minus 55 millivolt in mammalian axons. So here this impulse has to be equivalent to minus 55. So this will actually not generate any kind of impulse. So in order for an impulse to be generated, it has to, this excitatory signal has to reach up to a level of minus 55 millivolt. So now this level of depolarization, which is needed to generate an excitation potential, is called the threshold. So that excitatory signal has to cross that particular threshold, then only it will be able to generate a impulse. Okay, so now let's have a look at the action potential. So this is the resting membrane potential. So at this particular time point, if we look at the situation of the channels, there are some potassium channels which are usually open. Why? Because the potassium moves out, it is usually diffuses out of the cell. Now there is a response to some kind of stimulus. So in that stimulus, the membrane begins to depolarize. And now if that stimulus is good enough, which could brought the threshold to minus 55 millivolt. Now that threshold, upon reaching that threshold, there will be opening of sodium channels. So these sodium gates, they will open and there will be influx of sodium. Now this influx of sodium will lead to the depolarization of the membrane. So after certain time point, there will be, all the channels will open up, lot of sodium will be taken up. Now when this has reached to a certain point, or the spike has been generated, now there will be closure of the sodium channels and the potential channels will open. So in a way, this potassium will be moving out, that will try to balance. So in a way, this positive charge will increase in this increase or this depolarization because of the uptake of sodium. Now it goes down because of the flow of the potassium out. So this step is the repolarization. However, when this repolarization being attained, then at some point we see a hyperpolarization or the undershoot. So in a way, these pumps before closing, some excess amount of potassium ions they are moved out, which may lead to decrease in the membrane potential below minus 70 millivolt. Now that is called as the undershoot, as this hyperpolarization is below the resting potential. And after a few minutes, these channels, these potassium channels, they close up, and again we have the resting membrane potential of minus 70 millivolt. Okay, so these action potentials have two distinguishing characteristics. That is, they follow an all or none law. So it means that each depolarization produces a full action potential. So that reason is that the voltage gated sodium channels open completely at the threshold or they don't open at all. There's nothing like the graded events, or there's nothing like one opens, another channel doesn't open. Either all open or none will open. So if all will open, then there will be movement of lot of sodium ions inside, and that will lead to the depolarization of the membrane. Second one is the action potential are always separate events. So when impulse is generated, action potential is generated, and they are separate events. They cannot be added up like the graded potential. Why? Because after every impulse, there is a refractory period. So at that particular time, none of the voltage gated channels are open. There will be no movement of sodium, and that will lead to the no depolarization or repolarization phase. So we can say that production of an action potential results entirely from the passive diffusion of ions, and at the end of each action potential, the cytoplasm has more sodium and less potassium than at the rest. Why? Because sodium is taken in and potassium is thrown outside. So now in order to balance that imbalance of sodium potassium, now energy is required. So there will be requirement, and there will be active transport that could produce action potentials. Okay, so let's have a look at the how this action potential is propagated in an unmyelinated axon. So this is a typical axon. So here you can see a detailed one. So this is, we have the cell membrane, this is outer and this is inner side. So in normal time, normal at the arresting potential, the inside is negative, outside is positive. Now let's say the impulse is being generated. So in that particular instance, what will be happening, that sodium will be influxed. It will be taken inside. So that will lead to the conversion of this negative to positive charge. So as more and more sodium will be taken up, there will be more and more generation of positive charge, or in a way depolarization will take place. Now to counteract this depolarization, the potassium ions, they will be pumped out. So this potassium pumped out, and again the negative charge will be restored. So there will be a cycle of depolarization followed by repolarization. And this cycle of depolarization moves from one point to another. So sodium coming inside, potassium going outside. So that will keep on movement of the charge. That is, the impulse is being moved from one point to another. Okay, so in case of myelinated one, what we have is that these action potential, they are usually generated at the node of Ranvier. This action potential is not generated in the areas where the myelin sheath is there. So it can directly jump from the one node of Ranvier to the another node of Ranvier. So we call it saltatory conduction, or saltatory simple terms, it means leaping conduction. So that is the reason that the movement of impulse is fast in the myelinated axons as compared to non-myelinated axons. Also, the velocity of conduction is greater if the diameter of axon is large, or if the axon is myelinated. So myelinated axons, they conduct impulse more rapidly than the non-myelinated, because the action potential is only produced at the node of Ranvier. So a huge distance is just jumped in case of myelinated one. So let's have a look at this particular table here. So these are the different sites where we have presence of axons. This is the diameter of axons. This is myelinated or non-myelinated, and this is the speed. So here you can see in the giant squid, the diameter is 500 mm of the axons. So here we just take 25 milliseconds for the impulse to transfer. So in case of human leg, this is 20 mm, and here the speed is 120 milliseconds. So you can look at this size, the diameter of the axons. So the larger one is having the conduction faster and lesser time is we take. And if we compare with the myelinated and the non-myelinated one, usually in the myelinated one, the conduction is faster. Okay, so this is where I would like to stop for this particular lecture. So thank you very much for your attention.